Bah, j'y arrive plus là.
Ah bah, ça
J'ai
doit...
fait des oreilles.
Ok, merci. Bonjour à tous. Et en fait, je m'excuse, mais c'est mieux que je parle en anglais. En fait, c'est mieux pour moi et surtout c'est mieux pour vous, je pense. Mais naturellement, nous pouvons interagir en français. Je peux essayer de répondre en français, naturellement. Ok. Tout le monde peut écouter? J'ai un peu dans le micro. Ah, oui? Parfait. Okay. Today I decided to give you some examples of the work I do in cancer epidemiology from the point of view particularly etiology, and also a lot of people in our team, Générations and Santé, work on these topics as well. Okay. So just an introduction with a few slides to remind some key concepts, I think, what has been achieved so far, and in particular the fact that we know that around 40-45% of tumors could be prevented just modifying some key modifiable factors. Top priority, of course, tobacco. Explaining around 20% of tumors, and so on, diet, obesity, etc. And in fact, this information has been used already extensively by a lot of institutions, of course, and in particular my former institution in Australia, the Cancer Council, Australian Cancer Council Victoria, in very nice ads, education campaigns, etc. Like this one, don't be a statistic, with the different modifiable factors. And of course, tobacco is the number one. And of course, this type of activity has produced results, and in fact, quite striking results. And here is another ad from the Cancer Council Victoria. Smoking in Australia, in the 1950s, 50% of Australians used to smoke. Now the prevalence is 12.8%, quite a remarkable result. In fact, I should say that this type of result is not seen in most countries in Europe. This type of percentage is around half of the percentage, the prevalence in Italy, and I'm sure is probably half of what is observed here in France as well. And all these has produced, results in terms of outcomes. And the Cancer Council Australia here reminds us that lung cancer death rates have declined by 20% since 1990. Okay, so these are the achievements. So the question is, do we still have things to do as cancer epidemiologists? And what can we achieve further? So in the presentation, I'm not going to give you a systematic response to these. I just cannot. But I want to give you some examples of some clues about how to respond to this. Okay, so before showing these examples, I think it's important to remind ourselves what cancer is. And I suggest a book from Mel Greaves, a great book, Cancer, the Evolutionary Legacy. It's quite an old one, published in 2000. And there, of course, there are also more recent articles by him and his group that always remind us that we have to remind us of the evolutionary perspective to understand cancer. So you see here, the starting point is a normal cell. And from the normal cell, there are mutations, and some of these mutations are selected to allow the tumor to overcome barriers. And these barriers can be anti-apoptotic barriers, apoptotic barriers, anti-angiogenic barriers, the physical barriers within the tissue, and then so on. Further mutations actually to come to the barriers to distant metastasis, and further mutations allow the cancer to overcome these barriers as well and to become metastatic. So you see, it's really an evolution from the beginning of life, if you want, up to the diagnosing and spread of cancer here. And in a sense, and Mel Greaves reminds us very effectively, here we see an evolution during life, but in a sense we can see this also an evolution from the beginning of life back into the distant past, where in fact originally life was cells proliferating and proliferating. So in a life you start from a complex human being with all the checkpoints, and you might have to end up with life back in time. I think this is a useful, very useful model that I will show you repeatedly during my presentation. Okay, so the first example is about new tools to assess exposures for risk prediction and, in parentheses, causal inference. And I'll give you the example of DNA methylation. Some of you have already seen some of this work, and I apologize for this, but some of you might be interested in understanding a bit about what we have been doing on DNA methylation. So to remind you, what is DNA methylation is adding a methyl group to the cytosine. And so some of the cytosines in the genome are methylated and some are not. And what is the function of methylation of cytosines? I must say that we know a little about the function of methylation, but the classic model is that you have a gene and you have classically the promoter, and there is evidence, quite established evidence, that if the promoter is methylated, you read here, the expression of the gene is blocked, so the gene is not expressed. If the promoter is unmethylated, the gene is expressed. And what we know also in cancer, looking at cancer tissue, this is quite established. It is observed that we have two different features. The first one is CpG island, so we're talking about the promoter I was talking about before. We have hypomethylation of promoters in tumor suppressive genes. This means that the tumor suppressive genes is actually blocked. So, and the other feature is the opposite. It is hypomethylation of gene deserts, or regions in which there are no genes, and this goes to genomic instability. These are the two different features that we see in cancer tissues. So transcriptional repression of loss of tumor suppressive genes and genomic instability and mitotic recombination. So from the epidemiological point of view, what can we do with the biological samples that we have? And typically we have blood samples. And this is a study we have done in blood, looking at DNA methylation in blood and cigarette smoking. There are several, and actually hundreds, of CpGs across the genome, where methylation is very different in smokers and non-smokers. And these are all the different signals we see, these 461. These are p-values, but you can imagine this correlated with the effect size. Just remember this gene here, AHRR. There's a huge effect of smoking for this gene, and also this one here. And this is a zero, no effect. So first of all, you see that most of the effect is in the direction of hypomethylation. Okay. But a few genes like this one is actually hypermethylated. So this means increased methylation in smokers versus non-smokers, while here you have lower levels of methylation in smokers compared with never smokers. Okay, so This is actually focusing on one point, and it's the AHRR gene that I showed you before. And these are current smokers, these are never smokers, so you see hypomethylation in smokers. And also you see a nice trend if you look at former smokers according to the time when they quit. So these are people that quit quite closely to the time of blood draw, so they are in a sense more similar to the current smokers. And these are people that quit a long time before, more than 30 years, and they are more similar to the never. So there is a nice dynamic in what we see in terms of DNA methylation. Just in this particular gene, but the same thing or similar things we see in all the other 460 loci. This is and what I've shown you so far is in blood. This is in buccal cells. This is DNA from buccal cells, and we see very similar things. This is actually the correlation between methylation. This beta is an indicator of methylation. 0 means no methylation, 1 means methylation. Basically 0, no cell is methylated, and 1, all cells are methylated. In this particular locus there is again this gene AHRR. So you see the nice correlation between methylation and smoking pack years. The higher the pack years, the lower the methylation. So again, it appears to be a dose response effect. And it's interesting again to see this in buccal cells and to see this in also in blood. And this is an interesting example from a study in Auspach, and this is they looked at mothers and children and the offspring at different ages. They have repeated measures, and this is blood again. This is I really like this study that look at the different genes that I showed you before that are known to be associated with smoking. So methylation in HRR, it goes exactly in the same direction that we see in adults. These are kids, the blue one are kids of sustained smokers. So the mother was smoking at the time of birth. And you see that the methylation in the offspring is lower than the methylation in offspring of non-smokers. And this is sustained over time. Basically, this birth up to the age of 20. This is quite striking data. And you see the other, you remember the other gene, the MY01G. It was the one that had the hypermethylation in smokers. You see exactly that here. Hypermethylation in offspring of sustained smokers. So all these data combined are striking evidence of an association between smoking and DNA methylation. I'm not talking about effects here, and I'm very careful about doing that. We cannot conclude that there is a causal effect here, but the association is striking. So what we have done is then to look whether we can see an association between these particular loci in the genome, the methylation in these loci, and risk of future lung cancer, okay? In never, current, and former smoker, and also adjusting for smoking, and I'll show you in a second. So this is the classic Manhattan plot, like in a GWAS. We have the upper part and lower part because of two different studies. This is the study in Melbourne, and this is Équipe, Italy. And we have some signals, nothing that is nominally statistically significant. This is the plot of the two studies, the effect size. And so we decided to take those loci that have the lowest p-values and also that are consistent in the two studies. And we took that to replication in another 3 cohorts. And these are the results from the overall 5 cohorts. And we have 6 CpGs, so particular cytosines, in which methylation is associated quite strongly with lung cancer risk. And this is adjusted for smoking. So this is association independent on smoking. And we have a very low p-values. This one here, for example, is the one in the AHRR gene that I showed you before. So another question is, I was talking about causal inference. Can we say that this model is valid, or in other words, can we say that the effect of smoking on lung cancer risk, or part of the effect of smoking on lung cancer risk, is mediated by DNA methylation? It is very difficult to answer to this question for various reasons. On one side, we have a strong effect of smoking on lung cancer risk. On the other side, we have a strong effect of smoking on DNA methylation. One of the problems is that DNA methylation is also very different across the different cell types in the blood. For example, here you have mononucleosides, granulocytes, B cells, natural killers, etc., etc. And here is methylation, and you see that the different types of cell types show a very different methylation profile. So this makes the interpretation of the results difficult from the point of view of causal inference, because, for example, it could be that smokers have an inflammatory response that alters the distribution of cell types that may become different from never smokers, for example. And this may lead to then an association between DNA methylation and smoking. There are ways to try to estimate the distribution of the cell types using the DNA methylation data. But this is a quite crude way to try and adjust for the cell type distribution. And we cannot conclude anything definite on the causal inference side of things. Although I will mention Mendelian randomization, and what we are trying to do, in fact, is try to apply Mendelian randomization to test the hypothesis that, in fact, DNA methylation mediates the effect of smoking on lung cancer risk. But putting this aside for a second, I want to show you also the estimates by smoking status. So these are again the estimates for the 6 CpG sites strongly associated with the lung cancer risk for the never, former, current smokers. So you see that in particular the striking feature here is that the strongest effect is in fact in former smokers, actually systematically stronger than in current smokers. Here the problem with never is that the number of never is quite small, so the confidence intervals are quite wide. Although for some of the CpGs you see that there is a suggestion of an effect of DNA methylation on lung cancer risk even in never smokers. But this striking effect in former smoker, which is very difficult for us to explain, although I guess it is possible that if someone has quit smoking and the level of DNA methylation in these CpGs remains low, this might mean that the biological effect of smoking in these people have been sustained. And this might be an explanation, but at the moment it's still a speculation. But still, these results are important from the point of view of risk prediction. Because in fact what we have done is to look at the ability to discriminate lung cancer cases and controls using the 6 CpGs, the methylation of these 6 CpGs. So starting from the reference model, that is the classic model of risk prediction for lung cancer, it includes a reported smoking status, plus never form a parent, plus the number of pack years, the area under the curve is 79%. While when we include the 6 CpGs, the gain in the area under the curve is statistically significant but quite modest, 2.6%. But when we look in former, the increase is quite large, 11% in our ability to discriminate. And in fact, this could be quite interesting because one of the key questions in lung cancer at the moment is whether to use a CT scan for screening. And in fact, just using reported smoking is certainly not enough to make it sensible from the point of view of cost effectiveness. These results are encouraging in terms of potentially using DNA methylation for risk prediction and discrimination. Okay, so I want to just briefly mention a few things about genes. What can we gain from the study of genes? And I'm talking about the germline now, because in the model I showed you before, there's also the aspect of what we inherit from our parents, and therefore from the variants that are present in all cells, and therefore genetic susceptibility to cancer. And of course, we have been knowing for a long time that cancer runs in families, and there have been interesting results in the 90s with the discovery of BRCA1, BRCA2, and also in prostate cancer that I use here as a model. Recently, there has been quite a lot of excitement with results and discovery of this gene HOXB13 that comes out from a study in basically 4 families. These 4 families where people for years and years, even decades, have collected the information on families like these, and with recent technological advances, first linkage analysis and then the next generation sequencing have led to the discovery of some genes like this one, and a lot of excitement, of course. So this is the prostate cancer susceptibility landscape with HOXB13 that is associated with a relative risk of around 10, maybe 20. Initially, it was probably thought to be higher. And even BRCA1 and BRCA2 mutations in this gene lead to an increased risk of prostate cancer, although not as high as for breast cancer. But these are very rare mutations, in fact, and the utility of these is for a limited number of families, multiple case families like the ones that I showed you before. And probably the most important information from the point of view of the family, of the carriers, is not the relative risk or the odds ratio, if you want, but actually the estimate of risk, of their risk lifetime or at a certain age in the next 5-10 years to develop prostate cancer. And here is for different birth cohorts the estimate of lifetime risk here that we estimated actually using population-based cases around 1,500 in Australia, for which we had also DNA from relatives in the family. So here, this is important information if a genetic counselor wants to have this conversation with Mr. Smith carrying the mutation and want to know what's my risk of developing prostate cancer. And the genetic counselor with this data can say, okay, your risk is actually 40% in your lifetime, but it could be in fact even 80%, or it could be 20%. The problem here is the wide, very wide confidence intervals, in fact, which is not good for a genetic counselor when he has to give this information, and particularly for Mr. Smith, because then he has to decide whether to have maybe PSA testing every 6 months, every year. But then another problem is this associated with aggressive prostate cancer or not? Because with prostate cancer, the problem is that a lot of these prostate cancers are indolent and not aggressive, in fact. And in fact, the treatment might even be worse than the disease itself, in fact. And the problem is that, in fact, there's no evidence that these gene and the mutations in this gene lead to more aggressive disease. But there's also the side of things of the GWASs. And in fact, prostate cancer is an example that I've chosen because it is a cancer for which we know anything about the etiology in terms of modifiable factors. We know very little in terms of the high-risk genes because, in fact, for breast cancer we know we have many more genes. But in fact, it is the cancer for which the largest number of SNPs have been identified by GWAS, which is in a sense strange, certainly interesting. And here is a partial list of these SNPs, and there are more than 100 now. And you see the largest effect is with the 8q24. It was discovered some years ago and that we contributed to characterize. And then a lot of the SNPs have a very, very small association. But in fact, if you put together all the SNPs, and if you think about the genetic risk distribution, what often is called the polygenic distribution, you can see some interesting results. So here, the percentiles means this one here is the lowest percentile, basically men that have no risk allele. And this and here is men that have most of the alleles, that carry most of the risk alleles, of the 100 plus risk alleles. So if you consider this group as a reference group, you can say, oh, wow, there's a relative risk of 27, so there's a 27-fold increased risk in this group here. But in fact, it is the reference here doesn't make much sense. In fact, we don't know really the meaning of this group. It makes more sense to do something like this and to take the baseline at the average risk, which is the 25th. In this case, has been decided to choose 25th and 75th percentile. And in this case, relative to the population risk, basically, this top percentile has a relative risk of 4.4, which is anyway quite interesting. And this result has led people like Rose Hills and others to say that these could be a powerful tool to triage people into then further screening. Another problem, the problem remains what I said before, that most of even more the SNPs that I showed you here are not associated with a particularly aggressive type of prostate cancer. Okay, so then the third example I want to show you is that the use of genes to study the effect of modifiable factors on cancer. I'm talking about Mendelian randomization. And I want to give you an example of that. It's a BMI and lung cancer risk. In fact, many epidemiological studies, including this study using Équipe and Mutations, have shown an inverse association between BMI and lung cancer risk. So this is follow-up time, and here you see the inverse association. Then it seems to basically disappear, the effect here. So these might suggest inverse causation. Probably people with prevalent disease tend to lower their BMI, in fact. It is also possible that there is still residual confounding from smoking, even though these analyses are adjusted by smoking. So to test the hypothesis that there is residual confounding, probably from smoking, the people in Bristol, using data also from our Melbourne cohort, have done a Mendelian randomization study, in fact. That is basically using the large databases available with the SNPs and BMI information to identify the SNPs that are associated with BMI. Okay. And the beauty of this is the fact that the SNPs cannot be influenced by other factors such as smoking. So this is then to test the hypothesis that the genetic instrument is called, that it is the predictor using the SNPs, is actually associated with BMI. And this shows exactly that. So the genetic instrument, the combination of the, in this case, 97 SNPs associated with BMI, are actually associated with BMI and not associated with pack years or cigarettes per day. So then the trick is to see whether the genetic instrument is actually associated with lung cancer risk. This association will not be confounded by other factors such as smoking. And in fact, this is the results. This is overall. So clearly there is no inverse association between BMI predicted by the SNPs and lung cancer risk. And actually, by histologic type, this is adenocarcinomas, squamous cell carcinoma, small cell lung cancer. There's actually an increased risk for squamous cell carcinoma and small cell lung cancer, in fact. So this is quite a striking example of how to use genetic information to make causal inference, or at least to reduce confounding. Okay, and this is the last example I wanted to show you, and something we are focusing in particular at the moment. It is asking ourselves if we use tumor tissue, and in fact a lot of epidemiological studies, we have done this in Melbourne, we are doing this in Etrozen, are collecting tumor tissue, starting in particular from breast cancer, lung cancer, etc. The question is, so far, this has been done with the objective of taking into account heterogeneity in tumors. And in fact, this has led to the observation that different risk factors are associated differently with different tumor types. I'm thinking about, for example, breast cancer subtypes, etc. But there is more to that. In fact, we can use even more cleverly the tumor tissues. And I want to show you a study done by Mike Stratton and Nicholas Sanger Institute in the last years using TCGA data. If you don't know, TCGA is a large consortium funded by the NIH, NCI, to collect in a systematic way tumor tissue. In this case, the 2,000 tumors, were actually many more in the data set, which is public, in fact. These tumors have been sequenced and genomic, epigenomic data have been generated on a large scale. And 30 cancer types have been included in this large data set. Okay, so Alexandrov and Stratton and colleagues have used this publicly available data to generate these interesting results. So this first figure is actually the description of the 4 million somatic mutations they found in these tumors according to the different types. 30 cancer types. Here is pilocytic astrocytoma, so we are talking about a pediatric tumor. Melanoma here. These are the number of mutations, the prevalence in each tumor. Each dot is a tumor. I see two very interesting things here. The first one is the trend, an increasing trend. In a sense, it probably makes sense to have the lowest number of tumors in a pediatric tumor. Remind you that somatic mutations accumulate during life. It also is a normal process, and we know more and more about that. The other thing that strikes me is also the heterogeneity across a particular type. For example, these colorectal cancers. There's a huge variation in the number of mutations from one particular colorectal cancer to another one. But what they have done is also to look at the type of mutation, and in particular starting from the 6-point mutation, cytosine to adenine, C2A, C2G, C2T, etc. And then also they looked within each of these also the neighboring bases. And then they tried to classify these and trying to find patterns. In particular, they used something called the non-negative matrix factorization that is used also to take from pictures of the face to take out the particular features. And they identified actually 21 signatures. Here I've just shown the 14 of them. And then they say that this signature 4 is associated with smoking. And they say these on the basis of what has been known for a while from laboratory studies in which smoking causes particular C2A mutations. And then they also say that this signature 7 that is shows a lot of C2T mutations associated with the UV light exposure. Again, from previous laboratory knowledge that shows that UV light causes particular C2T mutations, etc. And this is the summary, basically, of what they found. Here, the 21 signatures, the different cancer types. So you see, then here the probable association I was talking about before. So again, smoking, signature 4 associated with smoking, they say. UV light, signature 7, and others. Many are actually of unknown origin. And also you see that signature 4 associated with smoking is actually observed in head and neck and all different types of lung cancer. So it makes sense. But for a particular type of cancer, and I've chosen here, this is signature 4 associated with smoking. I've chosen lung adenocarcinoma. Each bar is a particular tumor. You see that the signature 4 associated with smoking is prevalent in all lung adenocarcinomas, but there is quite a lot of heterogeneity and other signatures are present of really unknown origin. And the problem that I want to stress here is the fact that this work is based on TCGA data that basically does not contain information on the exposures. So the idea for us is really to try to adopt this particular paradigm, not necessarily the method, because there are also other methods that might generate different signatures. The limitation is the lack of information about the exposure. We could use this method applied on tumor tissue in epidemiological studies and combine this information with the exposure data, the extensive exposure data that we have to make some inferences. And I will just quickly go through these. I wanted to stress just the fact that, in fact, there is more and more evidence in the last 2 or 3 years that somatic mutations that are similar to those that we have seen in tumor tissues are present also in normal tissue. And this is just an article that I found in Science, published in 2015. This is a normal skin, actually, from the eyelid of volunteers, people completely healthy. They did sequencing on different parts of the eyelids, on the biopsies, and they looked at particular genes that are oncogenes or tumor suppressor genes that are considered to be prevalent only in tumors. And in fact, each of these are clones or subclones, different cells. You see most of the cells do not present any mutations, but many cells present mutations in these tumor suppressors or oncogenes. So our knowledge also about what is normal, and therefore the reference group is changing quite rapidly. And talking about these, about the presence of genomic abnormalities in normal tissues, I think this is a striking example. So this is a typical chromosomal translocation T1418 present in follicular lymphoma. And so basically means that this is chromosome 14, this is chromosome 18. Part of the chromosome 18 goes and attached to chromosome 14 and vice versa. So there is a translocation. And these basically cause a fusion of two genes. BCL2, that is present in chromosome 18, is fused with IGH, that is present in chromosome 14. And these leads to overexpression of BCL2 that is actually anti-apoptotic. So this is a very common feature of follicular lymphoma. And just concentrate on this pattern here. This is the frequency of the translocation in follicular lymphomas at diagnosis. Okay, you see the distribution, you see the quite high prevalence of this translocation. This is results from Équipe and Étiology is included here. These are healthy subjects. So this is quite striking because it is observable, the translocation also in healthy subjects that would not have developed follicular lymphoma. So there are, again, this is an observation that there are genomic abnormalities that can be found in normal people that might not necessarily lead to anything pathological. And these are actually healthy subjects that will develop, that have developed within Équipe follicular lymphoma after a number of years. So on average, there is a difference between these two groups. And in fact, these groups is quite similar in terms of levels of frequency of translocation with the follicular lymphoma at diagnosis. And this is just a validation of these results here in another cohort in northern Sweden. So really, I think our perspective of what is the process that leads to cancer is slowly changing. And we have opportunities, I believe, using DNA methylation, for example, as a marker of exposure, but also as a marker to predict more accurately risk. And I really strongly believe that the collection of tumor tissue and the analysis with the novel methodologies and ideas that are developed in the field of genomic medicine, etc., could be applied to epidemiological studies to gain insights into the etiology of cancer. And I concluded here and just acknowledging the fact that all that I showed you is the product of various groups and a lot of people that have contributed enormously to all this. Thank you.
Very nice talk. Can I ask you a question on methylation and what it means? So, as I understand methylation, it's a cumulative, it's a single cumulative exposure to something. Are there signatures that could tell you about the timing of that exposure? Because for cancer, there are a lot of, we know that there are these windows of susceptibility and exposure that may be relevant. So are there opportunities in trying to identify somehow when those exposures happen when looking at methylation?
But in a sense, the data on the former smokers suggest exactly that, in a sense that using methylation data, you can actually predict the time when they quit, and therefore it is actually the most striking and interesting aspect, I think more than the causal inference that is probably doubtful, is actually the fact that it is a more accurate instrument of exposure than self-reported smoking. Whatever detail there could be, whatever you've asked, the number of pack years, the duration of smoking, when they started, etc., it seems that the biological effect of smoking can be predicted more accurately with methylation data. The other aspect is that I showed you just the smoking, but in fact the methylation is being studied also for many other exposures, and in particular air pollution, for example, with some interesting results. There are some results also on obesity. There are a few CpGs that appear to be altered in terms of methylation in association with obesity. So I think that more than trying to understand the causal effect is more useful to really to predict risk, and could be a risk of various outcomes, in fact, not only lung cancer.
But I guess as a follow-up, but say you're really interested in exposures that happen in adolescence, or exposures that happen early in life.
Yeah.
How would that signature look, or is it possible to find? I mean, are there signatures that could basically distinguish between an exposure that happened early in life and later in life?
I cannot answer. For sure, the data from the HOXB, and there are others, show that methylation is a marker of exposure even in utero, potentially. The data that show that even after several years from birth, you see a difference in methylation in children that have been born from smokers or non-smokers. Whether it is different than between exposures in utero or in childhood and exposures in adulthood, I don't have data to be able to respond to that, potentially. Of course, to do that, you have to have repeated measures, measures in childhood, measures later in life that we don't have.
It could be an interaction with age, with methylation and age. That could be a hint that difference in nutrient profile with age.
In terms of an interaction, when you look at outcomes like lung cancer, yes, yes. Yeah. But again, to understand whether if you want to compare childhood and adulthood, the problem is that most of the studies that we have are studies in adulthood, and we only have blood samples.
The results for methylation in former smokers are very striking, because former smokers do have a reduced, I mean, the risk of cancer is not as high as in current smokers, so you wouldn't expect to see the same kind of association. So
exactly.
Is it possible that it's other exposure, that it's capturing maybe other risk factor? I mean, maybe it's a more global way to capture exposures that may have lung cancer.
It is possible. We have pondered a lot about these results with our collaborators at HIRC and Imperial. We don't have a plausible answer. Yes, it is possible that other exposures play a role, although smoking plays such a large role here, and other exposures play a smaller role. And it is such a striking difference that we still don't have an answer. I wanted also to add two important points. Is the fact that for what concerns the work on methylation and lung cancer risk, we have recently obtained a grant in collaboration with HIRC to actually do further analysis targeting the top CpGs, so probably not doing a genome-wide analysis, but on a larger scale, on 1,500 controls, and really with the objective of testing the ability to discriminate between cases and controls, in particular former smokers, and also to augment the cases in never smokers to see whether we see an association also in never smokers. That clearly is less strong, but still appears to be quite suggestive. The second point is that we also obtained My stagiaire, M2 stagiaire Anna Michessan, has obtained a fellowship from ENCA for a thèse, and she will work with me on this idea of applying the work of Max Stratton in the context of epidemiological studies. So we are quite excited about that, and we will start the work very soon. Okay, thank you.
Thank you.
Je dois faire